whether you're in academia or whether you're in a nonprofit organization. Medicinal chemistry is medicinal chemistry.
Yeah.
And whether you're in one therapeutic area or another therapeutic area, we all have the same goal, and we, in general, use mostly the same rules and properties, etc. And get into the CNS therapeutic area, you have to add in a couple additional parameters, which I'm going to highlight today. So if there's anybody in the CNS area in the audience, they'll know that there's yet another hurdle to overcome. Yeah. So I'm sure many of you have seen this before, but it is to highlight the fact that doing drug discovery is not trivial. And going from any assay development, if you're a biologist, hit identification, if you're in the biochemistry aspect, to doing high-throughput screening, to getting a pill into a person's mouth is a long process. It's a very expensive process. And here are some of the statistics shown here. You know, cost of drug to market really is, these numbers are old. I'm sure they've gone up to, you know, maybe a third more of that, whatever, but, you know, the odds are less than 10% of actually getting a drug into market. So, the work that we do at Southern, and clearly with our great collaborations with Ph.D., is really from the basic research to preclinical development, which I mean by that is identifying molecules that are tested in animal models and are showing efficacious results. This is to show once again the clinical candidate success rate. And highlighted are the bars here, which are blue for preclinical. Phase 1 is blue, and Phase 2 is pink. And really, it's the safety that's an issue, which comes into the toxicity, whether it's with regard to the molecule that you're moving forward, but many times also it has to be target related, as we recently have known. And blue is the pharmacology, meaning not showing efficacy, not getting into the brain, not being permeable enough to get into the active cells. This shows an undesirable trend and a disturbing trend, at least from a drug discovery perspective. The first set of bars is from 1996 to 1999, and you know, here we have, this is all for infectious diseases, cardiovascular cancer, and neurodegeneration. And you can see that as we proceed, this goes up to 2007, we're getting less and less clinical candidates getting to people. And we wonder why that is, and there's several theories, several philosophical views, and we can discuss those later. Once again, it shows the steps to get to clinical trials when you have a lead identification, which we'll talk about today, working with potency, selectivity, metabolic stability, how well the compound survives within the body, once you take it into your mouth, getting through the system, and then getting to the right target. Up to formulation, efficacy, clinical trial. This is, we always look a little puzzled sometimes, and we're always cognizant of the fact that we may be identifying intellectual property for UAB, for Southern, and we need to take that into consideration with filing patent applications. Guidelines to drug-like properties. So I'm going to go through these and show you the vast number that are taken into consideration in trying to move forward with an appropriate lead molecule. And then I'll break it down following this into what each of them mean. So the step 1 is the early stage. You have a hit molecule that has been identified for high-throughput screening, which I believe Bob Bostwick has already presented here. And these are some of the properties that we look into. I'll explain them later. Rule of 5, structural alerts, ease of synthesis, solubility, physical chemical properties of molecules such as log D, microsomal stability. This is hepatocyte stability in mouse, hepatocyte stability in human, IC50. And if you'd like a copy, you can use the slides. Try not to take notes. The second step is you identify your leads, you're going to optimize your lead. And some of the properties you look into at that point, so you have compounds that are shown potency, bioavailability, half-life, clearance, and then all of these are related to some form of toxicity, perhaps, and also getting into the target brain penetration in the red one, minimum effective dose. So these are properties that once you go into animals in vivo, how are they behaving? why aren't you getting the efficacy, or why are you getting the efficacy that you want. And then finally, you optimize those, you go into further preclinical development when you're going to identify an IND, so IND enabling studies. So genetic toxicity, in vivo studies, you're going to really repeat your in vivo studies, go in dose response, make sure you see a dose response. You do want to see toxicity so you can identify the safety window, the safety profile of the molecule. Biomarkers, if they're available, mechanism of action, the biochemists, biologists, entomologists are going to try to determine that, since that's key in going forward with filing an IND. And this is related here, I said, to AD drugs, but you want to show differentiation from a molecule that is now efficacious in your model from what's on the market. If it's a non-precedented approach, you'll want to show differentiation, say, to another, indicate another therapy that's out there for the same disease, or if not, then compare it to a similar type of prototype molecule. With CNS drugs, there's a separate ballgame here. In addition to all of these other steps, you're going to want the molecule to be smaller in size. We're going to talk about that. We look at the log D. We're going to talk about that. We want it in a certain range. Polar surface area is really critical for getting into the brain. And then you can look at the different proton donors, proton acceptors, rotatable bonds, how many arrow rings you have. It all goes into a balance of the properties, which we'll highlight at the end of the talk. So, most probably know what molecular weight is. It's the mass of one molecule in a substance. The polar surface area has to do with heteroatoms on a molecule, whether it's hydrogen, nitrogen, oxygen. Sulfur sometimes plays a role into this, but it's the surface sum of all polar atoms, including those attached to hydrogen. And it's used to optimize for getting into the cell, not necessarily being grade permeable. Solubility, this will come into effect if you have to have oral dosing. Compounds need to be soluble enough to be permeable enough to get to the site of action, whether it's peripheral or into the brain. Log D is, we use it more frequently than log P, although there's a lot of calculations out there for log P, which is a really good estimate in the early stages of drug development. But log D is the distribution coefficient, and it's a ratio of the sum of the concentrations of an ionized and an unionized form of a molecule. So if you have a NH2, it can be ionized to NH3, or if it's OH, it can be ionized to O minus. So it's taking into account all of the polar type of properties that a molecule can have. And they look at it in water and in a polar solvent and in an organic solvent, and the distribution of the molecule going back and forth into both of those phases. It measures the differential solubility of the compound between these two solvents because, when you're making compounds, they can go into organic solvents. But when you have to formulate a compound to be orally active, it's usually in a water-based solution. And then the log P is really just the ratio of concentrations of an unionized compound. So it's easier to reach the desired log P versus log D if you have more polar groups or ionizable groups on the molecule. And Lipinski's rule of 5, Lipinski's from Pfizer, and he came up with this rule of 5, I think it was in the early 90s, but he did a lot of research and realized that this is not only for CNS, for any drugs. Molecular weight is always, if you're doing small molecule identification, you should target less than 500. The C log P should be less than 5. Hydrogen bond donors, like NH2, SH, whatever can give hydrogen, should be less than 5 in your molecule, and then less than 10 hydrogen bond acceptors. So they're really stringent rules. These are just guidelines, and after a while you get kind of a feel for what's good and what's not. In the pharmacokinetic properties I mentioned, this stands for mouse liver microsomes, human liver microsomes. And this is a first pass metabolism type of quick and dirty test. You inject your compound into the microsomal media, and you look at what's remaining in the compound by HPLC, LC mass spec, whatever, after 1 hour or 60 minutes. And usually you want, at least 20% of your compound after 1 hour for early stage development. Ideally, you'll want it much longer. The liver microsomes contain, as I said, this is for first pass metabolism, metabolizing enzymes, CYP isozymes such as CYP1A2, 2A6, etc. It's widely used as an in vitro model system to investigate the metabolic state of the compound. However, there's always discussions, should we even do microsomal stability, because hepatocyte is really second pass. It's in liver cells, so it's really second pass metabolism. And we test hepatocyte stability after a 2-hour time frame on the compound remaining after incubation with the cells. This contains phase 2 enzymes, such as sulfatases, glutathiones, glucuronidation, which when you get into metabolism like this, it tends to be more toxic to the animals and more long-term effect. And so this is a quick read, but ultimately, if you want efficacy in animals, the hepatocyte stability is most present.
Is like the microsomal assay easier to do than hepatocytes, or maybe cheaper, so that's what you do first?
Yes, yes it is.
Okay.
So I think as you go along, as you get into more lead development, it's probably more preferable to just go to hepatocytes because you don't have as many.
Yeah.
So CNS drugs, as I mentioned, another hurdle for development is brain penetration. This I just want to highlight a study that we did at Pfizer probably in the early 2000s. We divided a bunch of drugs on the market into CNS indications, 26, and we had 56 non-CNS drugs. And these were for the therapeutic areas, and I shouldn't in here. These are just the names of the compounds. You may recognize a lot of them for CNS indications and non-CNS. So we were about 2 times more non-CNS drugs than CNS drugs. And this was from the top-selling drugs in 2003. And what we found, although there is a big error, the molecular weight in general is much lower for CNS drugs than non-CNS drugs. The C-log-P was similar, and the polar surface area was about 2 times less for CNS drugs versus non-CNS drugs. Now, what does Lipinski's rule say? He says the molecular weight should be less than 500, C-log-P less than 5, etc., etc. These results show that there's greater constraints beyond the rule of 5. Molecular weight should be less than 400, 2 or few hydrogen bonds, 6 or less hydrogen bonds accepted. So this is pretty lenient compared to what really is found with the drugs that we analyzed for CNS and non-CNS indications. The descriptors, as you can see here, hydrogen bond, acceptor, donor, rings, rotatable bonds, for CNS versus non-CNS. The results are they're about half the amount for CNS versus non-CNS. And there's no successful CNS drugs with more than 3 aromatic rings, and they're significantly less rotatable.
Were these drugs already shown to be effective for different neurological disorders, or were these just taken in order to study those?
Were they shown to be what for what?
Effective for all, like the CNS.
Yeah, so these are drugs on the market in 2003.
Okay.
Yeah, these were for CNS indications, and these are for non-CNS. So, for ideal properties of CNS indications, which is what our main focus was at that time, we came to see that smaller is definitely better. 300 is ideal, although that's really hard to get with everything. Polar surface area is 45, which is really hard to get, but I think around 70, for sure less than 80. And then, of course, the rotatable bonds and the hydrogen bonds. Now, structural alerts are called reactive moieties, or structural alerts is a term that was coined by some chemists. It's not necessarily a broad term, but I think if you did search it, you would find it. There's a philosophy to this, and one thing that many chemists will come to me when I say, oh, don't even look at that. Oh, but it's this. They'll say that There's compounds on the market that have that structural alert, and that's true. However, it's wondering what is the risk you really want to take? What is the effort you want to take? How much resources do you have? If you have a lot of resources, yeah, well, let's go for it, because there is that serendipity that always happens, right? But if you really want to play it safe and go to a path that you know you shouldn't have any effects, then avoid the structural alerts. They're associated with multiple examples of adverse in vivo outcomes. And I'm going to read this because I think it's really important. The chemistry, pharmacology, and toxicology provides a strong rationale for a grouping to be a structural alert due to the potential intrinsic reactivity, DNA intercalation, metal coordination, or metabolic activation to a species capable of covalently binding to biological macromolecules. Once you have a covalent binding, there's no going back. This binding could lead to immunogenicity, CYP inhibition, direct toxicity, carcinogenicity, or idiosyncratic, which is a lot of reasons why drugs are tricked from clinical development. Some molecules incorporating a structural alert may never generate an adverse toxicological or CYP inhibition outcome. And there are examples that include safe marketed drugs containing a structural alert. Attrition of a compound due to an adverse outcome without a recognized structural alert in a molecule, that happens. And a structural alert is not on its own predictive of an adverse outcome. Many other factors come into effect, such as clinical dose. I mean, you can have a structural alert, but maybe it's only needed a very low dose for a short time, right? Route of drug clearance or the presence of metabolic routes that divert metabolism away from the structural alert need to be considered. So, these are guidelines, these are things to be aware of. Doesn't necessarily mean to stop a molecule. So it's just using all the information you have to make the best test form.
So do you look at those in the hit list already or later?
I look at them in a hit list,
Okay.
and filters are done through the hits.
Okay.
And eliminate it. If you have a small list, then after you've done all these filters, then you could go back and figure out which ones aren't so bad. And let's move that forward until we see if we can get potency, at least with dose response or whatever, and then add that in.
Yeah.
So it's kind of taking everything into consideration, like with anything else.
Yeah.
This you won't read, but this is I carry this around like a Bible. I have two of them actually, different things. But what I want to show you is that they are real, right? And so these are compounds, structural types that undergo metabolic activation. These are potential DNA intercalation. So when people show me hits with 3 rings together, I wouldn't even look at it, but people get excited about this. So when I went up to Harvard, the first compound they showed me was a compound like this and saying it's lowering A beta. And I had no clue that, no, I didn't know that will give you toxicity. Let's see what else we have, So it's knowing, being aware of these things, and, if anyone ever needs any help or whatever, I'd be more than happy to talk to you. Alerts not relying on metabolic activation. So, these are straightforward, but then there are ones that have been shown to be metabolically unstable but aren't as obvious. And then metal chelators are over here, such as imidazoles, triazoles, pyridine.
Quick question.
Yes.
So should we eliminate if you are taking the lead hits, so should we just eliminate based on structure right away?
Yes.
If it is complex structure, then just don't.
Yes, because it depends on if you're going to do drug discovery. Yes. If you're trying to find a research tool or mechanism of action, then maybe not. Depends on how far you're going to take it. But ultimately, when you start with something like this and you have to modify it, you're not going to end up with the activity that you've seen there. And then you do need to be sure that the activity that you've seen is not due to toxicity. So whenever you run your assays, you should always run cellular toxicity assays at the same time. Yeah. The project, I'm going to highlight how we did some SAR work, some hit to lead for through a medicinal chemistry point of view is gamma secretase inhibitors for Alzheimer's disease. And this was the program, the key program that we did right before. This is just to remind you that the amyloid hypothesis of Alzheimer's disease pathogenesis starts with the cleavage of the APP protein by two well-known proteases, right? Beta secretase, gamma secretase. They form the oligomers, and then eventually goes on to form the plaques, which is clinical AD. The enzyme I'll talk today about is the gamma secretase. Okay. It's an intracellular enzyme, and it consists of 4 components that are well characterized, shown there: presenilin, nicastrin, alpha 10-2, and 2 of its substrates are ATP and Notch. When gamma secretase cleaves APP, it forms A beta. So when it forms A beta, then it can go on to form the oligomers, go on to form the or toxic species oligomers, and known to be toxic now. Or it can disturb the Notch signaling, which we don't want because then you get on side effects. So the profiles of the compound from a drug discovery aspect are: there's gamma secretase inhibitors, which decrease everything. These are 2 amino acids in the A beta sequence, 40 amino acids, 42 amino acids. 42 is more found in humans. 40 is a good representative to look at in animals and cells, etc. Gamma secretase modulators, which are harder to come by, they don't touch A beta 40, they reduce A beta 42, and they don't touch Notch, which you want it to be Notch selective. And then the Notch sparing gamma secretase inhibitors reduce A beta 40 and 42, and don't touch Notch. And that's the mechanism that we worked on. Clearly, the first type will show side effects, and the next 2 are preferred for drug development. The question is, and still remains, what is the required therapeutic window from reducing this A beta peptide and not touching the Notch? So, this is just a little background to show some medicinal chemistry that was on board when we started the program. Bristol Myers Squibb had a molecule. First generation molecule is shown here. They needed to improve the potency, and what they did was they tied up this here, this ring here, and they did different substitutions. Instead of a fluoro and an acid, they put this ethoxy group here. So they got down to 120 nanomolar, but here is the chiral center. And I don't know if people remember what chiral centers are, but at this center, for example, you can have a hydrogen going up and a hydrogen going down. But this data is a mixture of both, 50-50 mixture of both. Then they went on and they resolved it, so they got 1 enantiomer and they lost the potency. So that tells them then that the R isomer is more potent. They got it to 13 nanomolar. Then they opened up this ring because my guess is they were probably getting metabolic stability. I have this somewhere. And they got down to 4.1 nanomolar, and they got nice selectivity with Notch. However, it wasn't getting into the brain. There was low brain penetration. And in the rat liver microsome, the first pass metabolism was okay, not great. So what they did was they put a CF3 group. Fluorines are known to help a molecule get into the brain. They lost some potency. They also did tie up this free amine over here, this free amide. They lost some potency, but they pretty much maintained Notch selectivity. They wanted to bring back the potency, and all they did was put this magic fluorine group here, which happens sometimes. I've dealt with magic methyls, but not magic fluorines. And, you know, their potency increased substantially. And their Notch selectivity was okay. It was actually pretty good. However, as I'll point out later, this compound actually failed in clinical trials because of efficacy. Not enough efficacy and some toxicity, actually peripheral toxicity. Because 58 nanomolar is pretty potent, right? And so whatever the dose is, they had to lower the dose to not see the activity, but when they lowered the dose, they didn't see the efficacy. And so they're just some of the things that Bristol Myers did with their compound. This is a Wyeth compound here. This is more easier to see. It's not very potent. It has this primary alcohol. Primary alcohols can get glucuronidated in second-pass metabolism, form a toxic species. Gets glucuronidated, it usually stays glucuronidated. Anyway, they wanted to improve the potency, and they did, and all they did was add this ethyl group here, and methyl group here. Selectivity is tenfold, or notch. They even improved the potency more by replacing this phenyl ring with the thiophene, substitute chlorothiophene. However, what they found was they got oxidation at this methyl group, glucuronidation here. They're still pretty potent, but it's very unstable because of these metabolisms. So how are they going to make it more stable? They put CF3 groups on this dialkyl chain, and they did improve it in mice. Human, they did not. Remained potency, remained as selective. Stability-wise, what they did was they removed this methylene group here to make it just closer to the site of attachment, so less spots to get metabolized. And they improved it, metabolic stability. This compound was not selective enough, and they saw extreme toxicity and was not. So the issues were shown here, as I highlighted a couple of them: the non-selectivity, the brain penetration. I'll show you later some profiles of the BMS compound where it's not soluble and molecular weight is over 500. And so the conclusion was, when we see these clinical failures, improper balance of the compound properties, there's always discussion about are the clinical trials designed appropriately for Alzheimer's disease. And then did the preclinical pharmacokinetics, dynamics, and animal data support the dose and the design going into the clinical trial. Okay, so for our program and for any program, you need a screening tree. How are you going to push hit compounds forward to get optimized lead molecules? And it can vary extensively for any program, any assay that you have. Should they be cellular? Should they be functional? Should they be enzymatic? The first one we used here was an enzymatic assay, purified gamma secretase, which is a pain. We had a purified enzyme, isolated, set it up, but we got it into a pretty high throughput mode initially while we were identifying hits from the initial hit, which I'll show you, to get new lead series. And we get these compounds that look pretty potent, meet our criteria. We want to get initial physical, chemical, pharmacokinetic properties about them. Are they soluble? Are they stable? Whatever. Not necessarily killing the series, but prioritizing the series. Based on resources, too, we had a really small chemistry group. So if we had a group of 10 or 20, maybe we could work on them all, but we had to really make a lot of smart decisions all the time, and it was very tiring. After we get the series identified, we go into stealth. We use A-beta 42 in the non-cellular assay and the cellular toxicity assay. And, you have your series identified. Now, this is in cells. It's not only acting gamma. So when a compound was going forward, we'd always go back to the enzymatic assay to make sure it was hitting our target and reducing the gamma secretase. So active and interesting compounds coming out of our cellular assays. We do a preliminary PK again. And they moved down into in vivo candidates because we wanted to get brain penetration, brain permeability, PK studies, maximum tolerant dose to go into animals. And we go into the mice to get our proof of principle. This can go, if you're concerned about the molecule, you want to get as much data, depending on if you're, who you're presenting to, what package you need. You can get your CYP inhibition selectivity panel up here as well. So I don't think we really go back and forth. We had some side assays also, and it can vary for whatever program, from microdialysis studies to see if you're actually reducing A-beta in the brain. Neuronal culture assays we also did. And I'll explain to you why we had to do that. This is just, in general, you want a compound profile for any program. Whatever your, these are some physical chemical properties, And then these are the pharmacokinetic properties. And you'll want to have guidelines. These guidelines don't change for any program. You know, less than 15% remaining is low, the intrinsic clearance, 20% or more you want after 1 hour, 20% or more you want after 2 hours in the hepatocyte. Solubility doesn't change, or the same for whatever indication. Polar surface area, you want to be tuned into that for CNS. Molecular weight. So I mean, in general, it's all the same. But you do want to have, you know, your selectivity and your enzymatic activity or cellular, whatever will be particular to the program you're working on. This shows you that medicinal chem is an iterative process. So you make compounds, they look good, you test the potency, the PK goes back. It doesn't meet it, it goes back to making more compounds. Compounds that meet your criteria go on, test in vivo, or not, terminate the series. It's a way, the iteration is a way of prioritizing your series as well as eliminating them, whether it's even a series or a program. All right, so gamma-secretase chemical matter. We had a couple of leads. One of them was from this paper published by Greengard, and he was into Gleevec, cancer drugs, but he noticed that Gleevec reduced A-beta in cells, and he also had this other series came forward, was by a Pfizer group actually, and these pyridoprimidines were claimed to report A-beta in cell reduction. We did, before I came up there in Harvard, they bought a bunch of compounds from Cochras, 50 compounds, and they tested it in the A-beta enzymatic assay, the gamma-secretase assay, similar to this, and what they found were these two leads. This is where my laboratory called lead started. We had a lead that was 60 micromolar with this alpha-beta unsaturated ketone, total structural alert. This is a Michael acceptor. Any negative charge atom that comes in is going to form a bond here and stay. It's a covalent bond and stay. And then we had this 1367 compound from Cochras, and it was 20 micromolar. I said, wow, these are great. But what attracted Dennis Upwell and Mike Wolf, who founded my laboratory, was that these compounds were showing notch selectivity. And 5 or so years ago, that was really key in the fact that compounds have failed since then, that they can get compounds that were showing potency in an enzymatic assay right at the gamma-secretase, but they were really showing notch selectivity. So that's what prompted them to form the drug discovery group to try to improve on the potency of the molecule. As I pointed out, this was reactive, and this actually cleaved. It was very unstable. It's reported in the literature actually as a kinase inhibitor to form this. So we had to fix that compound. In the meantime, so we did. We looked at, particularly this. We tried to repeat this data in our assays, and we could not see any reduction in beta. So then we actually made small pieces of it, metabolites, and we couldn't get any activity either. We went on to look at these pyridoprimidines. These became aryl-amides, and these, when we reduced this ketone, became these alcohol type of molecules. So at that point, after 600 analogs, what we found was that the amino alcohols, the notch selectivity was there. We couldn't really improve the potency. Aryl-amides, problem with potency. Pyridoprimidines were our first series where we got under 5 micromolar activity. We maintained selectivity, but they're unstable and cytotoxic. So what this allowed us to do was get our biochemical assays fine-tuned for the drug discovery effort. We got our infrastructure established for sending out our compounds, process all in place. And we decreased our in vitro screening concentration from, I think it was 100 micromolar to 50, and we were down to 10. So we thought, well, 600 compounds will work that way. As we were going on, as we were coming to the conclusion that that was an issue, a common med chem activity is replacing amides with sulfonamides. And so we did replace it with sulfonamide. The conformation, potentially, it's a different, it's an sp4, different center of orientation. You'll get different binding activities. We also converted the naphtha to aryl and alkyl benzylamine, so we had a lot more diversity. We can make a lot more compounds quicker, as I'll show you. The synthesis is really short. And we did improve. So the sulfonamides, we went from micromolar activity to nanomolar in 40 in vitro, the gamma secretase, in cellular activity, natural activity. So we knew we were onto something. And then the three chemists all went onto the sulfonamides. This is just to show you. So another part of MedChem is you don't want more than 10 step synthesis. You can't, it's not, eventually when you go into clinical development, it costs a lot. However, the flip side of that is if it's the only drug for that indication, then it's worth it. But it's very hard to make kilos of compound, in particular, depending on the number of steps, the reagents used in the steps. So this was so attractive for drug development. It's basically 2 steps, in addition to if you have to make some of the starting materials, which were pretty easy. So what was the max? 4, maybe 5 steps. Nice. We'll want to always have a checklist for whatever program you're in of the chemical matter that you have and the lead attributes that you have. And they're all the same: potency, structure, activity, relationships, cellular activity, selectivity, safety, physical, chemical, all those properties I talked about. Intellectual property is key. If you're writing a grant application, you should always mention in there whether you have it or not that you are aware of it. That, because ultimately we all strive, I think, in one form or another for making drugs that matter. And, you want to have a patent application that supports the property for your institution. It's really fun. We went on to test compounds in animals, obviously, and through the iterative medicinal chemistry process, the things that we needed to look at, and one will always have to have properties that they need to focus on, was we need to block A beta production, we needed to have natural activity, we didn't know the window we needed to have, and we were targeting to have improved properties of our molecules that were better than the failed clinical candidates at that time. Because if we needed to license, we can only carry compounds so far. We're going to need to find a partner to help us move our programs forward. And so they'll always say, well, what is better? Why are you better than BMS? Why are you better than Wyeth or Merck or whoever else was there? The drug-like properties that did differentiate our compounds were brain penetration, the size of the molecule, log D. So we were doing really, really good. This was busy, but it's important. It's the first set of compounds for wild-type mice. I'm going to highlight 2 of them on the next slide so you can see them better. But really what's in the red is the selectivity of the Notch ELISA. Actually, we used first a Notch luciferase assay, which is a reporter assay, everyone knows, and doesn't show you the direct activity, but it does give you, usually sensitive, gives you good indication. In red, so here is BMS, the acetate, has a lot of red, and mostly in the stability, liposomal stability, hepatocyte stability. Polar surface area is 114, went on to phase 2.
Wow.
Molecular weight, 521. It failed though.
Yeah.
So really it's 0.17. So I think sometimes compounds get on a road in industry and they don't stop because they're looking good and they have the money, so they might as well try it. It would be a first in class. You have to balance all those. But so what we saw was, here's the luciferase versus ELISA, A beta ELISA, and our compounds were really quite selective. And so we picked these. Well, this one went into transgenic mice. These we went into wild type. I'm just going to talk about these. They looked pretty good. This one at the end here had microsomal stability red, but we thought, well, this is really potent though. This is 8 nanomolar. This is 120, 160, so it was kind of a spread, different profile. We didn't know what profile was going to translate into the animal study. This set is the same set of compounds, but we then started to look at the Notch ELISA assay. We developed the Notch ELISA assay rather than a Notch luciferase assay after we had started the animal studies. And if you see, the selectivity is not good. So it was really disheartening because the fact that these profiles look pretty green. This is important, I think, because I know a lot of luciferase assays are run, but it was an indirect reflection of the Notch proteolysis, whereas the Notch ELISA assay was a direct measure of the selectivity. The one thing that we wanted to do as well, and I'm not sure if I'll say this right, but was to get the same amino acid sequence that we used in the A-beta ELISA assay as we did in the Notch, because we're using a different cell line. But it was closer to the real thing than not. And when we saw the selectivity issue, we said, oh, you know, it wasn't a good thing. But we did see activity in animals regardless. These are the compounds I highlighted. This was the compound that was really potent, but not very metabolic stable, and it was 12% reduction. And so this is the reduction in the A-beta 40 in the wild-type mice. The best compound was this 1231 and then 1130, which is they're similar in structure. The only difference is this ring on the bottom is cyclohexyl. Here's the BMS compound, 16%. These were 3-month-old males, PO, delivering 100 mg per kilo. There's different philosophies of that. You know, you go high to see an effect. If you see an effect, then you cut back and try to do dose response. We wanted to see an effect, we needed to find a partner to develop our compound. So if we can show that this chemical series did what it was supposed to do, there was a path forward. We gave it 3 times a day as well. The 2 compounds, the one that showed 12% and the one that showed 40%, the difference from a med chem standpoint is this is a cycloalkyl with a fluorine, this cycloalkyl with a methyl. And here was where really it broke down, was the microsomal stability and the hepatocyte stability. So first and second path. We did med-ID, and I'll show you. It is a cycloalkyl ring. Methyl groups will always get oxidized, and OH will come in and oxidize them. Depending on what it's attached to, it'll be more or less. We went on to, trying to figure out what to do with the methyl versus fluoro and the cycloalkyl, because the cycloalkyl ring brought in a lot of potency. We went on to explore the right-hand piece, the acid piece, and we made a lot of different amides off of that acid. This is the second generation of compounds, and you'll see that they have the cycloalkyl methyl. We thought maybe it would not be as unstable if we added something over here. Cycloalkyl methyl, this is a phenyl. We had selectivity. So remember with the ELISA before, we were only at 100, which wasn't as good as BMS. But here we were getting better selectivity. In particular, here we were 404, and we thought, well, that's better than 100, and we'll have to show long-term toxicity studies and all. But it's a path forward. But from here down, everything's red. We said, oh my God, So we're getting 2013, and we said, well, how are we going to improve this metabolic stability? Or should we test in animals, IP sub-Q? And I really hate doing that because ultimately it's an oral indication. But, our livelihoods are at stake, and so we thought, well, let's try to test those IP and sub-Q. We had gone, as all this is going on, we did the metabolite ID of 1138, which is this compound that has a cyclohexyl methyl. This amide, that's the one that has a 400-fold selectivity with the Notch ELISA assay. And wow, what did we see? We saw a lot of metabolites. Basically, the parent was, all they identified was a total of 47% metabolites, but the main thing was really interesting was that it had, the whole cycloalkyl group came off. Even though it's 4%, it was a little bit surprising to a medicinal chemist. Obviously, you get oxidized. This is a soft spot, and then you get dioxidized both on the amide side chain and on the cycloalkyl methyl ring, and then you also get double oxidation. And so we did early SAR studies on the cycloalkyl ring of putting in a heteroatom, because sometimes adding a heteroatom to that will stop, but we lost our potency and everything. And so should have this program had gone forward, we would have worked harder at that soft spot. This I don't expect you to see, but I want you to understand what has to go into selecting compounds, for example, sub-Q or an IP, and looking at all the different indications like the Tmax of how long a compound is in the system, the Cmax, how much is there, the Tmax time, the half-life. I mean, these are the IP and sub-Q in the plasma, the concentrations, and this is the IP and the sub-Q administration in the brain. And so using all these parameters, we had to select compounds to go into animals with these routes of administration. What we found, I'm just going to highlight these 4 here because that's the parameters I just showed. We didn't see a decrease, significant decrease in A data, and we even saw a slight increase due to the issues with the pharmacokinetics of the molecule. And we had to go through a whole different formulation plan for the compounds as well, not all the same for the IQ and the sub-Q. But we wanted to show, because we were at the end of 2013, to outside interested parties that these compounds were truly gamma secretase inhibitors, and we went into neurons, cultured neurons, and here no pharmacokinetics are involved. And the compounds, we picked 2 prototypes, and we did see. So the first line is in neuron cells, the second one is in the the CHO cells, and we did see actual, reductions in the neuronal cell culture. So we knew that they were doing what we wanted them to do. And this really was microdialysis. We did another tool to show that they do reduce the A beta and A beta 42 in the brain, even though when we did it in mice, we didn't see the reduction.
And the microdialysis was direct injection in the brain?
Yes. ICD. So an overview of the lead compounds, we had these benzoic acid analogs, which were active in mice, orally active, stable, soluble, but not very potent and not better than avagastat selectivity, BMS compound. These amide side chains, this is where we got really the potency, and these substituted arrow compounds, they were more selective and they were very potent, but they were insoluble, metabolically unstable, and in the human body. This is just an overview. All chemists will show you the things that are good. Mono aromatic ring on this side. Bicyclic rings were no good, the positive and the negatives. And so out of the 1,400 or so compounds we made, this is what we get. So obviously the right balance of compounds are important. It's not always the most potent compound. It's not always the most selective compound. It has to be just right to give you the optimize whatever type of compound you're going forward to give you ultimately the proof of concept POC in animals. And so, we made it farther up to here. At the time, we tried to sell our program very hard to some interested parties outside, but we were up against 4 failed clinical candidates with this match toxicity issue, and the industry and the AD portfolio went to base inhibitors. They went back to base inhibitors, the other enzyme at the early stage of the hypothesis. Years ago, we did base. I worked on base. That was trouble with PK. They were in small molecules. They were peptide-like in nature. Went to the gamma secretase, now they're back to base. So we couldn't sell the program last those times. However, we do need to make this bridge happen, in particular for many indications. Here I'm talking specifically about Alzheimer's. This, I just want to show you Southern Research's chemistry department, the capabilities for medicinal chemistry. Any compounds that come as hits from the high throughput screening that need to go through filters, whatever. We have all of these capabilities, computational chemistry. We're hiring a crystallographer. We instituted an in vitro ADME group. We have, of course, NMR's exact mass to characterize our compounds. Parallel synthesis, intellectual property is important. Things that make it through all here go into in vivo pharmacology. And, this is a lead development optimization to get to the proof of concept to making drugs that matter. And here is just a sampling of some of the programs that we have in collaboration with ADD, UAB, broken down into disease areas, broken down into grant types, and then the project, and then the status that we're at. So we're quite busy. Right?
Yes.
And with that, I'd like to thank you. And this is my contact information. Mike, of course, has it. If there's anything that you ever want to just call the question up, I'd be more than happy to help. Because it's my life, you know. Part of it. Good part of it.
Any questions?
In all those leads, the slide before this, you showed, how many of those do you have industry or other kinds of investment, outside investment?
Oh, I don't. You can help me here.
Well, for ADD, we don't have any yet.
Of these, no, because they're actually early
They're very
stage.
early.
So they're all lead optimization, which means, you know, we have a good series, we're
Pretty much
optimizing.
internal.
Yeah, so we would typically go to an outside party to see if they were interested in licensing, and the IP that we have, after you show proof of concept in an animal model, they typically won't be interested before that. Everybody you talk to, especially with some of the really cool targets that we have in our portfolio, like TXNIP for diabetes, or Taufin for Alzheimer's disease, everybody says, call us when you have in vivo data. But you can't get good in vivo data until you have all those properties dialed, because you need to have the PK right, the potency right, the selectivity right, and so on. So it's even a long ways away from getting something to work in your mouth, longer than some people think. It's a lot of fun. For some of the projects we work, we meet every 2 weeks, just to see, you know, what's the newest data on the newest compounds that they're making. You can really see the iterative process in action, where, you know, they made new series of compounds, and all of a sudden half of them don't work at all, or they completely lost their activity. So it gives them more and more information about, we can mess with this part, but we really should keep this triazole here. Internal terminology. Thiopine.
Yeah, I think the ones that have medicinal chemistry really start, well, LARC is well along with MedChem. The TXNIP is coming along, Taufin, 1450 theta. I mean, we've actually had
CD38.
CD38.
TGF-beta.
Yes, I mean, there's a lot of
Well,
chemistry that's been ongoing with them.
Yeah, I have a preprint of the LARC inhibitor, if it's the right one, you have to show a proof of concept in rats.
Andy has showed proof of concept with the Pfizer compound.
Oh, so that's not
No,
the
not-
ADD yet.
No,
Yeah.
no,
Okay.
it's not.
We're trying to beat Pfizer compound. Yeah. So we have a good baseline. And the patent population, now we could make our own Pfizer compound to compare.
Uh-huh.
Yeah.
All right. Well, thanks for coming. Next week, we're going to talk about how to submit an IND.
Oh.
So much fun.
I'm gonna take care of some things that's going on.
Thanks so much. I loved the examples. That was really cool
Oh,
to show
good.
kind of the
Yeah.
progression.
Sorry, I interrupted in the middle.
So the,
I'm Patsy Oliver, and I'm working on the week
His compound.
now.
You want to do the same thing?
Yeah,
as
hey,
the
it's
Pfizer
the chemist in
compound.
my group.
Right,
Oh,
yeah. So
So
I'm working on
I've
the series for
the first batch was
November,
now
November,
8.
so
Yeah,
we'll do that.
Pfizer and good toxicity.
Uh-huh.
Clear.
Yeah.
And also
I got
Pfizer
another
compound.
last week, as you know, yeah.
I tested
half of them,
Uh-huh.
I'm gonna use the
Yeah.
other half this week.
So
And then we ordered,
kits
to do,
order and like
our
our
olive
Oh, good.
So this is a whole new area for her, so we can show, you know, that they're going.
We were waiting on some results actually this week.
Yanni is done, he does.
If it all looks
the
good, it's going to go in the
pancreas,
animals.
pancreatic, and we did the. He
You
just,
know, a few
I
months.
just
Like
put them
the
in the
summer.
pancreatic. I'm doing pancreatic right now. This is the B. Hi, nice to meet you. He's
I guess
done a little bit with.
It
I guess you learn not to get too excited
Yeah.
about it.
But,
Yes.
Yanni did,
Temper your enthusiasm.
flash with the first
You
test.
need to stay excited
You know,
because
the
it is.
ones that he found
You also-
activity were also,
Corinne
fortunately,
has
the
a
ones
good
that I
job.
found activity. So, we're gonna try.
Well,
to
yeah, there's 1,600 compounds.
Specificity question pretty hard because we've got a grant deadline coming up, so I've got stuff on the way to, try to look at some of that other pathway. So it'll be interesting to see